So I just want to welcome Dr. Aubrey Gray. Some of you are familiar with the SENS Foundation and his work there. He's also known for earlier work, I believe, was with the Methuselah Foundation. And we just caught him actually on his way to London. Is that right? All right. So Aubrey Gray, thank you.
Thanks very much. So it's more than 10 years since I've been in Vancouver. So it seems to be working. Oh, I already have one. Yeah, fine. I'm not entirely at my best because I got up at a ridiculously early hour for no reason whatsoever because my flight was delayed. I apologize on behalf of United Airlines for the mechanical fault that meant I'm 2.5 or 3 hours later than I was supposed to be. But here we are anyway. I know that a lot of you are familiar with the work that I and SENS Research Foundation do, but fear not, because this will be a very unusual and new talk. For a couple of reasons. First of all, because I'm speaking after J.L. Shansky, and I thought it would be a great opportunity to make fun of him and generally debunk a lot of the smoke that he blew at you earlier on. But also because I have some genuinely material stuff to discuss. So I apologize for a second time because this talk will be somewhat unpolished. It is the first time I've given it, and so we'll just see how it goes. But luckily, I believe I have 50 minutes. Is that right? So anyway, I have actually rather few slides, but anyway, we'll see how this goes. So first thing I want to emphasize is that I'm not going to be distinguishing between life extension and healthy life extension. I think there's far too much is made of this distinction. And the reason that not much should be made of it is, of course, because they are very, 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 very closely linked. The number one cause of dying these days, by a very large distance, is not being very well. Okay, so the fact is, if you're healthy, you're not going to die anytime soon, probably not. And if you're not healthy, you have a higher probability of dying sometime soon. So there is no point in distinguishing between life extension and healthy life extension when we talk about the intervention in aging. Of course, this is only to a first approximation. There are certain interventions, certain changes that have happened over time that have marginally changed the difference between those two numbers. Because we make more success in postponing causes of death that kill you quickly and less in causes of death that kill you less quickly and so on. But it's a really, really minor thing compared to the overall grand scheme of what's happened over the decades and over the centuries. So, before I start really getting going, I'm going to ask you all some questions. I'm going to ask you 4 questions. I'm going to ask you what you think is the current state of affairs in respect of 4 key statistics with regard to human longevity and related things. The first one I'm going to ask you is life expectancy. This LEB says life expectancy at birth. And I'm talking worldwide here, average over the entire world, all countries, developing, whatever. Okay, what do you think it is? Hands up anyone who thinks it's in this range, between 55 and 60. All right. Next, 65, okay. Next, 65 to 70, that should say, I'm sorry. Okay, next, then 75. All right. Hold that thought, because I'm not going to tell you the answer until the next slide. Remember what you said, all right? Next, fertility rate. So the total fertility rate, that's the number, that's calculated in rather the same way as life expectancy at birth. How many children the average woman is expected to have? Worldwide, again, average across the world. How many people think it's in this range, between 3 and 3 1/4? Or indeed greater than 3 1/4? Yeah, right, okay. How many in this range? How many in this range? And how many in this range? I note that J.L. Shansky, who ought to know more about this than almost anybody else in the room, is being particularly coy about putting his hand up. I just thought I ought to point that out. All right. Age, okay. Average age of everyone who's alive today. What do you think? Is it between 15 and 20? What's the question? The average age of everyone who's alive today, worldwide. Anyone who thinks it's between 20 and 25? 25 to 30? All right, 30 to 35? All right. And finally, proportion of deaths this year, or let's say last year, that were caused by aging. So let me be careful about what I'm defining this to be. So there are various statistics out there. There are various ways of classifying causes of death. And, there are, you can have big classifications like non-communicable causes, communicable diseases, accidents, injuries and so on. Okay. Within those categories, of course, you can subdivide. You can say which particular cause, and some of them are more age-related than others. So you can come up with a pretty good answer. I mean, if you want to come up with a really precise answer, you have to be a bit subjective. But if you're looking at this kind of level of accuracy, then it's pretty easy to say that, well, pretty much all non-communicable causes of death are related to age because they're chronic diseases that are progressive. Some accidents, some injuries are because of falls. Some infectious diseases are because certain infectious diseases are easy to fight off if you're young, like influenza, for example, and less easy if your immune system has gone downhill because of aging. So one can come up with a pretty good number here, to this kind of level of accuracy anyway. So, question is, what proportion of deaths worldwide are caused by aging? Is it this kind of number? Is it this kind of number? This kind of number? This kind of number. All right. Hands up anyone who didn't put their hands up for any of those questions. All right. Now, so I told you to remember your answers, so make sure you do. Here are the actual true answers. It turns out that the life expectancy of birth worldwide went over 70 about 5 years ago. About 2009, 2010. The total fertility rate worldwide dipped under 2.5 about 7 years ago, around 2007. The average age of everyone alive is not quite 30, but it's very close. It may get over 30 this year, otherwise it will certainly be next year. It's 29 point something at this point. And the deaths from aging are probably going to go over 75% next year. Those are the numbers. So, hands up anyone who got all those numbers right. Whoa! Someone actually claims that he got... I wasn't watching you. All right. All right. Hands up everyone who got all those numbers wrong. Yes, quite a few more. All right. I won't go any further. But you get the idea. I think it's actually really important for everybody who is interested in this topic to understand what's happening. To understand how rapidly things are changing, where we stand on matters like this. And the amount of ignorance about this, even among people whose profession it is to know about it, is profound. This needs to change. We need to have better information, better attention paid to what's going on, what the situation is and how rapidly it's changing. All right, but here's the bad news. I've told you some good news. Everything I've told you so far sounds wonderful. We're improving things really, really well. But things are bad. I think one of the things that Jay probably told you in the last talk, probably before I arrived, was that life expectancy has been increasing quite rapidly over the past while. But what he probably also told you, and what I agree with, and which a number of other demographers claim is not the case, is that things are slowing down. Things are slowing down quite badly, in fact. This is probably the most important statistic. Because this talk is new and I haven't really been able to get all the right slides together, I don't have the right slide that I would like to show you, but this is a remarkable thing. This statistic comes from the UK, actually. It came from last year, if I remember rightly. About 15 years ago, some demographers took what was going on, what had been going on for the previous couple of decades, and they made a prediction about how many people over the age of 100, centenarians, there would be in the UK in 2010 or 2012 or something like that. And it turned out they were wrong by a factor of 2. And they were wrong in the bad direction. We had half as many as you would have expected. We seem to be hitting some kind of wall. If you look at supercentenarians, people who are actually over the age of 110, it's even more overt, more clear. The oldest person who's ever lived, verified anyway, Jeanne Calment, whom we've heard about from Jay, to 122. She died 17 years ago. What the fuck? You know, why haven't we done better? Why haven't we exceeded that by now? What's going on? It's worse than that. We haven't even come close. The second oldest person ever died 3 years younger, about 119, around the same time, I think, a couple of years later. Generally, the oldest person who's alive is only 115 or 116. You know, we seem to be really hitting some big wall here. We need to take this seriously. The news that you tend to hear about longevity going up is not the whole story. There is good news, but there is also bad news. So I want to talk about that, and I want to talk about it in a realistic and constructive manner. And I'm going to start here. A lot of you are not biologists, I bet, but I bet that pretty much everybody here has at least high school maths. And therefore, I'm going to give you a bullshit-free version of how to do demography using high school maths. I'm going to tell you why I think that the pessimism that Jay often described as optimism is actually very misguided. And I'm going to start by looking at survival curves. So you've seen some curves like this in Jay's talk. And you've seen a bunch of other curves as well that were drawn in different ways. Things like mortality rates drawn on a semi-log scale, things like that. Lots of other ways that demographers very correctly use to give good quantitation to what's happening and what they think is going to happen in the future. But I think it's extremely useful to understand that one can eyeball survival curves, the kind of curves that we see in the general media, and get a good feel for everything, a pretty good feel, a good enough feel for everything we really need to know about what's going on during the progression of medicine and life extension. It all comes down to these equations, which are very familiar to demographers and which I think probably came up earlier. Back in 1825, an English actuary named Benjamin Gompertz noticed that, to a reasonably good approximation, the risk that people had of dying in the coming year went up exponentially with age, at least if you ignored childhood and infancy. And so he came up with this equation. There was some kind of equation like this, where you can have a coefficient here called A, that is the intercept, often called the initial mortality, but that's very misleading, as I will explain in a moment. And then you have the slope, B, and this equation gives you the mortality rate, the mx, where x is the age, where someone's age, the risk of death at that point. About 35 years later, in 1860, another actuary named Maycomb, whose first name I forget, what's his first name? Excellent. Now there is realized that actually we could do better. We could characterize a wider range of mortality rates with age if we did it this way. We added an age-independent term. We just said that the mortality rate is the sum of this exponential term and this completely age-independent term. And what I want to emphasize to you now is that we can look at survival curves and we can see what these things actually mean without converting them into this actual equation. We can eyeball survival curves like this and we can see what's going on. The age-independent term is basically the slope of the mortality of the survival curve in youth. The more horizontal this is, the smaller that age-independent term C is. So back in the 16 or 17 or 1800s, it was like this, and later on it was much more horizontal because C had become smaller. If you only look at the recent things, when you factor C out, then you can also do the same thing for A and B. First of all, you can say that A is the length of this line. If you start at 50% mortality, you say, how long do half the people live? If basically this is dominated by this term, that the smaller this term is, the longer this line is. The larger this term is, the shorter this line is. And similarly, for B, the Gompertz slope, as it's called. the larger B is, the steeper this line is, the more close to vertical it is. And this turns out to be quite a useful thing, because it means you can eyeball survival curves and figure out what's really happening and what has happened in life. And I'm going to do that for a little while on this slide. What happened between the, let's say, 1700s, before medicine really existed, and the mid-1900s, the early 1900s anyway, was what people often call rectangularization. And that was more or less all about reducing this number, C, the age-independent term of the Gompertz-Makeham equation. It was turning this line into this line, something that was more horizontal and up here. Of course, I'm ignoring infant mortality here. The Gompertz-Makeham equation does not pretend to talk about what happens in very early life. So forget about what's going on there. Just look at the slope. The slope of this line is becoming more horizontal. Another way to extend life, in principle, would be to reduce this number, A, the Gompertz intercept. And that would involve shifting the whole curve to the right, basically extending this line. And a third way, in principle, to extend life would be to reduce this number, the Gompertz slope, B. What that would do is what I'm showing with this arrow. It would take this line, which is nearly vertical, and it would stretch the whole thing outward. It would move it to the right and also change its slope. That's what the changing of these three parameters does to these survival curves. And of course, this is all pretty approximate, but that's fine. All you need to know is a sense of what this all means. So, what's actually happened? There have been two eras in the past of life extension that have happened. Very little indeed happened until a couple of hundred years ago. But then, a big, big thing happened. We realized that hygiene was a good idea. We figured out not long after that how to develop vaccines and antibiotics and stuff like that. And what that mainly achieved was a reduction in this parameter, the age-independent parameter of the Gompertz-Makeham equation. It did this thing. It reduced C. And that caused this rectangularization. You can see what I mean by rectangularization, right? The survival curve here is kind of triangular at this point, and here it's not exactly rectangular, but it's much more close to being a rectangle than it is to being a triangle. So that's the origin of the term rectangularization that you will often see in literature about this kind of thing. But, of course, there's only so horizontal that a line can be. Once the line is completely horizontal, or very nearly so, you can't make it any more horizontal. And therefore, you're going to hit diminishing returns. You're not going to be able to increase longevity appreciably further by any further reduction of this number C. You've got to do something else. And by the mid-20th century, that was pretty much over in the industrialized world. Certainly in the Western world, it was a little later in Japan. So we have to ask ourselves, and people obviously did ask themselves, why have we continued to see a very respectable rate of increase of life expectancy in the Western world in the past few decades? And the answer is, we've succeeded in reducing A. When I say 1800s here and 1900s, of course, what I'm talking about is not the actual eras during which it happened, but rather the eras during which it began, during which the technology to allow it to happen occurred. What really seems to have happened in the late 1900s, and still going on, is that we've seen this right shift, this reduction of A, even in the absence of any opportunity to further reduce C. We have seen this right shift of the survival curve that has occurred because we have been able to do things like improving infant and even prenatal nutrition, reduce smoking, cohort effects, as they're often called, that have allowed people to essentially be biologically younger throughout their life than let's say, their parents were at the same chronological age. And so you can see that this is actually what we're having. These lines here around the 50% mortality thing are not getting any more vertical, they're just moving to the right. So this is all established. I haven't said anything remotely controversial so far. I've just put it in as non-technical terms as it's possible to do. But now, let me actually go a little further. This here is a phrase that Jay Olshansky has used many times over the past several years. He likes to say this. It's one of the more exasperating things. And he says a lot of very exasperating things, but this is one of the most exasperating. He actually said, everyone who ever said that we can defeat aging has one thing in common, they're all dead. All right. Now, I'm using this term up here, MRDT, that stands for mortality rate doubling time. It's just basically the inverse of the slope that I was talking about earlier. So that's what we're talking about. And the reason I mention this, and why I call this the MRDT challenge, is because what Jay actually means when he says this is that... Hang on. Is this. He means that we have failed to change the Gompertz slope. We have failed to change that number. I'll go back a couple of slides. We have failed to do this. We have failed to change this slope here that I've been talking about earlier on. If we could do it, as we might, in principle, be able to do in the future, then this would happen. And that would be the end of Jay's prediction. What Jay is telling us about the past, which is absolutely correct, is that we have not made this change. When he says this, he is not departing from my view at all. It's absolutely correct. The question is whether it will continue. Now, Jay is a historian of science, among other things, and so I thought I would tell him a little bit of history of science and tell him something that his grandfather said in 1900, which resembles... So really the question is, do we have a prospect of actually entering the third era of life extension? Having essentially finished the era of reducing the Macon term, the age-independent contribution to mortality, and having largely completed what we can realistically achieve in terms of reducing the Gompertz intercept, the rightward shift that I was talking about earlier, can we realistically look at the only remaining alternative, reducing the Gompertz slope? In other words, increasing the mortality rate doubling time. That is what I'm going to spend the rest of my time discussing. What might do this? In 1993, Elizabeth Corder at Duke published a really seminal paper showing that one of the major causes of sickness and, of course, mortality in the Western world was predominantly, well, not predominantly, but was greatly influenced by which allele one carried of this particular gene, apolipoprotein E. Turns out that within the human population there are 3 alleles, 3 genetic variants, and one of them you don't want to have. For whatever reason, these variants are not called variant 1, 2, and 3. They're called variant 2, 3, and 4. Don't ask me why, but they are. And variant 4 is the one you seriously, badly don't want to have. It turns out to be extremely bad for you in terms of your, the age at which you're likely to get Alzheimer's and also the age at which you're likely to get atherosclerosis. Obviously, these are 2 of the most important sources of ill health and death in the Western world today. When this was discovered, I think it's fair to say that pretty much everybody predicted that it would be the first in a nice steady stream of similar cases, of other genes that would be discovered that had substantial influence, where genetic variants had substantial influence on one's risk of ill health and death in old age. Turned out that didn't happen. Years and years and years went by, and no other cases were discovered. In the past few years, there have been some cases that have been discovered. And we understand why it took so long, and the answer is in the nature of what has been discovered, namely this here. Tiny, teeny tiny effects. The possession or lack of possession of a particular allele of a particular gene other than ApoE in the human population confers like small single digit, like 1 or 2% difference in one's age-related risk of ill health and death. It's teeny tiny. and very uninteresting, really, if you're actually not a biologist or whatever. I believe, and I think this is now also fairly much the consensus, that the reason why any such gene as ApoE can exist is because of this thing, antagonistic pleiotropy. That's what AP stands for. What antagonistic pleiotropy is that where a particular genetic variant has a benefit and also a deficit. It's good for you and also it's bad for you in some different way. And therefore, there is some kind of balance of selection over evolutionary time that maintains the heterogeneity in the population. The best-known example of this that was discovered many, many years ago is sickle cell anemia. It turns out that there's a particular variant of hemoglobin where if you have one copy of this variant, then you are protected against malaria. But if both of your copies of that gene are this mutant copy, then you get sickle cell anemia. So it's kind of maintained in the population. The situation with ApoE is still somewhat more uncertain. There are plenty of hypotheses out there, but it looks like it's the same kind of thing. And the thing is, this doesn't happen very often. Most genes do not have a pleiotropic effect. They're either good or they're bad. But a big, big, big thing has been happening in the past year or two, which not many people know about yet, and so I want you to know about it. People have been looking harder at really rare variants, rare alleles, rare genetic variants of genes. This hasn't happened before. If you go to 23andMe or any of these things, they will tell you plenty about what situation you're in respect of certain genes, certain genetic variants. But the ones they will tell you about are what are called SNPs, single nucleotide polymorphisms. And something that many people don't necessarily pay attention to about single nucleotide polymorphisms is that the only things that count as SNPs are things where the minority version is present in at least 5% of the population. So if there's a particular variant that only 1% or 2% of the population has, you won't find it from 23andMe. You won't find it from any of these analyses. There are plenty of things that are present in in more than 5% of the population, so there's a lot of variation, and that's why SNPs are useful. But not everything is like that. And it turns out that that's rather important, because when a mutation happens for the very first time, of course it's present in an infinitesimal proportion of the population, and it may randomly expand just by luck or whatever for a while, but if it's basically bad for you in an evolutionary sense, then it's going to be eliminated fairly soon. So it will never reach the level of being a SNP. However, evolution has different priorities than what we do. Evolution cares about the longevity of genes. We care about the longevity of people. Very, very different. So it is abundantly possible that there may be genetic variants that turn up here and there, and they're eliminated from the population pretty quickly because they are, on the whole, bad for you from an evolutionary sense, but which are good for you in other senses, especially in a modern civilized environment where we have medicine and so on. And it turns out that this is not just theory. These things are turning up. Just last year, actually 2 years ago now, the group in Iceland called Decode, who work on the Icelandic population, identified a rare genetic variant in the gene that encodes the amyloid precursor protein, the protein that gives rise to senile plaques in Alzheimer's disease, that appears to protect really powerfully against Alzheimer's. And sure enough, in the intervening time, that allele's protective capacity has been verified in other populations. It's a big effect. There's a lot of noise about another type of amyloid at the moment, an amyloid that happens in the heart, which is driven by a different protein called transthyretin. The reason there's a lot of noise about it is because it seems to be extremely important in killing really old people, centenarians, supercentenarians. Turns out, again, there are protective alleles that were discovered only in the past couple of years. And the reason this was discovered was actually because there are also damaging alleles, which are also rare, which accelerate amyloidosis in the heart. And if you have one of those nasty alleles, but you also have the protective allele, mysteriously you don't get the problem. So this was the first way it was discovered. There's going to be a lot more of this, I strongly predict. We are going to see a lot of this. So now, let us ask ourselves some questions about what we could speculate, what we can infer from this in relation to the survival curves that we see. In this kind of community, there's a great deal of interest, as we all know, in the extreme tail of longevity, in the longest-lived people. And I mentioned already that it's a little bit curious that the world record longevity hasn't changed for 17 years. Jean Calment, aged 122, is in no danger of being overtaken anytime soon. Sarah Naus, who is the runner-up, aged 119, same deal. If we look at the possibility that the way you get to be very old is not just by being lucky in a large number of ways that a lot of people are lucky in a few of, but rather being lucky in a small number of ways that extraordinarily few people have any of, then you've got a different kind of mathematical conclusion. It could be that in order to live to even 110 or 112, you've got to have at least one of these really rare protective alleles. And it could be that if you've only got one, then you just aren't going to get past 116, 117. But if you've got two, then you might get to 120. Now, if that's true, and if there are actually lots of these out there, but they're so rare that it's just unheard of to naturally, by accident, get more than two, then maybe there's a lot of fertile ground here. Maybe with the big, big power that we have today to analyze genomes, we may be able to identify a lot of these things and figure out how someone could actually live a lot longer. I'm not saying even 150, but maybe 130, maybe 140. The kind of numbers that are at the absolute edge of what someone like Jay would predict to be possible. And that's awfully important. All of you know that what I work on is rejuvenation. I work on the development of therapies that don't depend on what genetics you have, on repairing the damage that the body does to itself. But I am very interested in ensuring that as many people as possible are able to benefit from the therapies that I'm trying to develop. And there are two ways to do that. One is to develop those therapies as soon as possible, and the other is to find bridges to them, to find things that we can implement sooner that will help people to stay a bit healthier for a bit longer. And this could be a very important component of that. So my last slide talks about how that might be. Everything I've said so far revolves around genetics. It revolves around what you were born with. And those of you who were not born that way, you may be thinking, well, oh dear, I wasn't born that way. But that is not the whole story. We are not just talking about genomics here. We're talking about transcriptomics as well. We're talking about what those genes do. The genes, the transcripts and the proteins that those genes express. We're all about damage repair late in life. We're interested in rejuvenating people who are already in middle age or older and taking them back to early adulthood. And the tools and the options that we have for doing that are exploding. It's insane what's happening. And a lot of those tools are genetic, not just in terms of manipulating our DNA, but also in terms of manipulating gene expression. One of the most appalling things, one of the most tragic things about the nature of gerontology today is that almost everything that's done to try to actually explore intervention in aging is done on animals that are in the lab and you do the thing to them starting either before they're even conceived because they're transgenic interventions, or at least you start when they're in, weaning or whatever. So we have extraordinarily little data with regard to what we can do late onset, starting in middle age or whatever. But some of what we might be able to do in terms of manipulating our transcriptome, the gene expression patterns of our cells, may actually achieve a degree of rejuvenation, maybe even a large degree. In other words, some of the things that the most extremely longevous people like Jean Calment had going for them may be things that we can apply to people who, for the first 70 years of their life, did not have those things going for them. We don't know. We cannot know until we try whether that would be the case. A key thing we need to understand is that we are not talking about epigenetics here in the way that most people talk about it. Epigenetics is a terribly, terribly fashionable field. People are terribly interested in the fact that people's gene expression is different at different ages or in different disease conditions or whatever. But almost everything that we are learning about epigenetics is with regard to adaptation, with regard to what the body already knows how to do to minimize the deleterious effects of aging or whatever. That's not what I'm talking about here. I'm talking about very obscure epigenetic features that are going to be pretty hard to unearth. But I believe, as I said on the previous slide, that we're getting now to the point where we have the sheer power, the sequencing power and the computational power to be able to extract information from this that makes me much more optimistic about the relatively short term, maybe 5 or 10 years from now, than I was even 2 or 3 years ago. So I'll stop there and I'll be happy to answer questions. Go.
Have those two ladies that you mentioned that lived very old, have their genomes been sequenced yet?
My understanding is that neither Jean Calment nor Sarah Naus had their DNA isolated and sequenced. The only person I know, the oldest person I know who's been the subject of a really thorough study was a Dutch lady who, you can remind me if I'm wrong, Jay, died at the age of 115. So that's pretty good. But I think we definitely need those two people.
Can't that be done posthumously with hair or something?
Certainly, but of course there are permission aspects.
Of course.
So I'm curious, with your organization, with this new thing that you're discovering, is this mainly coming from you in a perspective of a positive attitude towards an observation you're making, or are you in the process of starting to consider what your foundation can do with regard to this?
Great question. I don't yet know. Of course, we're very small. We have a budget of only $5 million a year, and at the moment, we have absolutely no scope for expanding the breadth of our work. But I certainly feel that this work is high priority. I think that, in fact, it may not be we who are best placed to do it. There may be other people, even Longevity Inc., Craig Venter's group, for example, who may be in a better position to do so. So this is really an idea that I'm putting forward for the community in general, with no particular aspiration that Sensory Research Foundation will pursue it ourselves.
What do you think the possibility of finding these genetic variants would be? It seems kind of like looking for a needle in a haystack.
Great question. So how do we go about this? How do we actually find really rare genetic variants? Why are SNPs defined to be things that are present in at least 5% of the population? Answer, of course, is it's all a numbers game. Absolutely. Now, short answer to your question is some of it is indeed serendipity. The transthyretin example I gave, basically what happened was they got lucky because this particular one gene, transthyretin, happens to have a lot of mutants that are inherited that cause accelerated accumulation of amyloid in the heart of the people, and that's why it's been a recognized disease for a long time. So people just looked at these families. It's an inherited disease. And they saw the curiosity that there were a bunch of people who were siblings and who carried the pathogenic allele, but they didn't have a problem. And so they asked why, and they actually had a look and found that a lot of these people had their homologous gene, the other copy, not the standard copy. And so they hypothesized that this must be protective. And then they did a clever thing. They went and looked for that putatively protective copy in the general population. In other words, looking at people who didn't have a bad copy on the other side, right? And sure enough, they found a massive increase in longevity, in life expectancy. So this was a kind of lucky guess. I don't actually know the full story of how the the amyloid precursor protein protective allele was found. I think it was just because it happened to be a little less rare than normal in Iceland, which is, as you know, the population that Decode have focused on in this extremely intensive way. But ultimately, it's all about the more data you have, the more chance you have of finding more ways to get lucky.
Aubrey, in looking for the kinds of people that carry these semi-adaptive mutations, is there any way, to your knowledge, to identify these kinds of people before they've lived for an extremely long time? Is there any data to suggest that people that perhaps look extremely young for their age, when they are in their 30s or 40s, that's not solely as a result of their lifestyle over other people's, avoiding certain toxins or bad habits like smoking, or that we might actually be able to include them in the cohort of people that we're searching for these rare alleles in?
So there's some scope for that. But, of course, what one always runs up against in that kind of line of thinking is the inaccuracy and imprecision involved in any kind of biomarker of aging, whether it be cosmetic appearance or whether it be something in your blood or whatever. There's been, as you probably know, I'm sure you know, an enormous amount of work gone on over the decades looking for reliable biomarkers of aging. However you may define that. Let's suppose you define it as predictors of how long someone's going to live or whatever. And it's not been very, very effective. I think the more promising way is going to be retrospective analysis. And the fact that we're mainly thinking here in terms of genetics makes retrospective analysis much more practical, because it means that one can do, as many people are doing, one can look at the families of people who have already lived a long time, their kids, their grandkids even, and thereby get some kind of correlation.
It seems that some problems can be framed in many ways. So Jay approached aging by taking that curve and scaling it. You're looking at aging by a different parameterization. So that makes me wonder, how many different ways can one parameterize aging, and how does that affect the approaches one takes to deal with it?
Okay, so I didn't see the beginning of Jay's talk, and so I'm a little bit handicapped here, but I strongly suspect that there is actually no difference between the way that he and I parameterize things, and Jay's nodding. It's just that I clarified and highlighted what Jay's really saying when he expresses pessimism with regard to the future, namely that he's saying that because we have not historically made any difference to this one parameter called the Gompert slope, the chances are we never will. I think he accepts absolutely that if we did, then that would be synonymous with actually doing what he says isn't going to happen. Right? The question is whether we can. And that's why I think it's important to focus on this. About 10 days ago, I gave a talk at the Gerontological Society of America meeting, at which I gave a few of these slides. In particular, I focused on that aspect, and I focused on, actually, maybe I have even the, yeah, here it is. This is something that I focused on as a set of things for discussion with regard to how we might study markers of this. A lot of work, as I just mentioned in response to the previous question, has gone into trying to find markers of longevity, biomarkers of aging. But different people mean different things by that phrase, biomarkers of aging. At the end of the day, most people kind of mean how long you're expected to live. So in other words, markers of future life expectancy, remaining life expectancy. And really what I'm trying to say is that in the human population, that's the wrong thing to be looking at, because we've pretty much done all we can when it comes to pushing out the longevity by means other than changing the mortality rate doubling time, the slope of that Gompert slope. So maybe what we can do is we can go to our existing data, let alone data that we might gather in the future, and actually analyze it in a different way. Look for genetic correlates, for example, with mortality rate doubling time. actually subdivide the population according to possession of this or that allele, and say, okay, maybe this population lived with this Gompertz slope and this population lived with that Gompertz slope, that might give us some hints that would actually be instructive with regard to the thing that Jay thinks is impossible.
We'll just take 2 more questions here.
Okay. I actually have 2 questions. First, but I can do one if you want. Anyway, you said your SENS Research Foundation is aiming on doing stuff for people later in life, and I was wondering what your plans for deploying these therapies are in real-world situation.
So we do think about
population.
So we do think about that. Of course, we are a biomedical research charity, and therefore our main focus is to develop these therapies in the first place.
Or rather, your fantasies for how you want it to happen.
Yeah, I got it.
Okay.
So the bulk of our work at the moment is in cell culture and in mice, but absolutely, our goal is humanitarian, and so we want to join the dots. We want to make sure that these things are not just developed, but also implemented and disseminated. And some of our work is actually focused on that. Just very recently, we completed a study at the University of Denver with a group that have a great forecasting system that has been developed over about 30 years. It's used by the UN and so on. We funded them to extend their system to be able to look at a scenario in which aging was brought under medical control and to look at all the things that people make scare stories about, overpopulation and so on, and to show that these things are basically nonsense. And that it's highly likely that these problems won't occur, which is a great thing to take to politicians and policymakers and opinion formers and get people more on side and less scared. Similarly, we're interested in licensing. We're interested in regulatory affairs. It's extremely bad news that it takes so long to get a drug from clinical trials out to the public, and a lot of people are interested in trying to accelerate and lubricate that. We're projects at Oxford working on that. So yes, we're working on all that stuff.
Okay.
Do you guys research on herbs and natural supplements?
No. We think that while it's valuable to stay healthy, the enormous majority of what you need to do to stay healthy is what we all know already. Don't get overweight, don't smoke, have a reasonably balanced diet. If you're averagely healthy already, then our belief is that the amount of incremental benefit that you can get from esoteric things, from exotic things, is tiny. It's not negligible. It's not zero, and I certainly don't say don't do it. I also don't say drink as much as I do. I mean, I happen to be well built in that regard. I can eat and drink exactly what I like, and nothing seems to happen. But that's not what I recommend. The point is, yes, do these things. Yes, do whatever you think works for you. Pay attention to your body and do what works for you. Don't believe any books, because books, by definition, are the lowest common denominator. They try to tell the general audience what to do, and people just differ from each other too much for that to make sense. So the best advice is pay attention to your body, do what works. But the number one thing you have to do is never be too complacent about how much benefit that's going to give you. It's going to give you a teeny-tiny amount, and therefore the only way you're really going to live a substantially larger amount of time is by giving me large amounts of money. Are we done? Sure, no problem. One more.
Otherwise, we're going to be setting up for the panel up here. Just want to give Aubrey a big round of applause there. Thank you for coming out.